at two levels: the fertilization stage and during the
Okay, I think there's a problem with Dr. Pavia's connection right now. Uh, I think he mentioned a while ago when we were testing broadcast that it was raining in his area, so I really don't know. Yeah, he lives up in Antipolo, so maulan daw doon. So I think it has affected his um, uh, presentation or his internet connection. So I do think we'll wait for him. I think he'll be reconnecting soon enough. But while having this short break from the Choral Talk by Dr. Pavia, we would like to thank our sponsors. So the 13th Annual Scientific Conference of the Philippine Society for Developmental Biology is in cooperation with Macare Medicals Incorporated. And we would like also to thank our other sponsors, Merck, MedTest, Medical Test Systems Incorporated, and Phil Anaserve Incorporated. So muli po maraming salamat po sa mga sponsors namin. And I think Dr. Pavia is back. So sir, welcome back.
I got removed from the meeting. So I'm almost done. So apologies for this. So can you see my screen now?
Yes, sir.
Yes,
Okay,
nakita po.
so there are two ways by which we can do this. First is the shotgun style, like I said, where we just allow these special colonies to interbreed um, between species, of course, and then that's the first level of hybridization, and then between the same species. And of course, the juvenile colonies will be further fused together to form the best possible combinations, but still it's natural recombination. And then the next would be the directed or the assisted style, where resistant to bleaching. This is a fast grower. We collect the egg and sperm bundles, and then we really manipulate which gets fertilized. And then based on that, we can further fuse the colony. So it's a more streamlined and directed approach, of course, which is harder to do. Now, I leave you with these pictures, because up until now, we can't have pictures of chimeras. People are still thinking, what would a chimera look like? And that's one reason why I want to do this study. The aquarium trade is the main source of pictures of coral chimeras, because in the wild, corals normally are brown in color because of the zooxanthellae in them. Many of the pigments are hidden by the zooxanthellae cover. But in aquaria where the light is correctly controlled and nutrient levels are kept to a minimum, and the coral doesn't need to have a lot of zooxanthellae present, the pigments of the corals really shine through. And you can see, not people have been observing chimeras for a long time already. So for example, you have this plate coral where you have two separate mouths and two separate colors. Normally it's a solitary coral, but you see these two organisms come together and fused into a single skeleton. The same is true with this other plate coral. And then here we have a euphylid coral. No, it's either a herpolithid coral, where you can see two different colors of the tissues present, but they live in one skeleton. And then another, for example, the Montiporas, they're very popular in aquaria, and they've recently observed multicolored corals. And for the longest time, they were saying this is the result of intense lighting, or they're just variegated strains. But people are starting to observe that, for example, if you break off a fragment from the green part and make sure there are no red tissues present, it's going to grow into a green colony. If you take a fragment from the pure red part of the colony, it's going to grow into a red colony. But if you get a fragment with both red and green parts, then it grows into an organism like this, where the red and green are interlaced together. So this is what I was telling you. We can observe it in large polyp corals and or those with very clear pigmentation. I would really want to study the real time distribution of the various genotypes, because that will spell the difference whether the chimera is able to function well. What use is a chimera if you have a big colony, but there's a chance that one of the genotypes will only be present in 10% of the colony? If the other half dies, then it would be pointless because the entire colony will go with the remaining 10%. So that's why it's important to study the dynamics of the fusion interactions of chimeric corals. Now, to end, I leave everyone with these thoughts. The effects of climate change on coral reefs have been studied, determined, and predicted through rigorous scientific inquiry. The effects on coral organisms have also been determined. In preparing coral reefs for the effects of climate change, the preservation of the reef structure is as important as the survival of the coral colonies themselves. It is no use to save the corals if no coral reefs, the large reef structures, are left. Habitat is as important as the component organism. Genetic variability has been observed within coral species. The creation of chimeric coral colonies serves as a dual function of producing larger colonies in a shorter period time frame and also producing multi-genome coral colonies, same species, that can survive and persist multiple stressor events. Now, the repressed, undeveloped immune system of juvenile coral colonies across many species serves as this very important link in the creation of these coral chimeras. Now, I'm a big fan of creating chimeric corals within bounds, but I will admit, before anything thinks otherwise, I believe that chimeric corals are not the only way that coral reefs can be preserved or prepared for climate change effects. It is but one tool that can be used together with other established and novel techniques to improve the survival and persistence of coral reefs in the midst of a changing planetary climate. We need a multi-level, a multi-discipline, a multi-practice approach in order for the reefs to get the best protection that they can. That's why experimentation and a questioning mind are very important in preserving coral reefs. Thank you very much.
Maraming salamat po sa inyong presentation, Sir Ricky, about these Franken corals. Diba? You mentioned these coral chimeras and how can they be used to preserve corals and maybe study resilience and immunity or immune responses in these animals. So, sir, kindly stay for the question and answer portion later after our next presenter. Maraming salamat, sir. Okay, so once again, that was Dr. Richard Thomas B. Pavia Jr. And his presentation on coral chimeras. Our next presenter is still going to talk about coral. So this is actually more on coral conservation. And our next presenter is Dr. Jonathan A. Anticamara. And Dr. Jonathan Anticamara is a professor at the Institute of Biology of the University of the Philippines in Diliman. Okay. Okay, and he's also the Ecology and Taxonomy Academic Group representative for the Research Committee of the Deputy Director for Research and Extension. He is also the curator of, is one of the curators of the UP Biology Invertebrate Museum. Outside the University of the Philippines in Diliman, he is also the lead author in the Intergovernmental Science Policy Platform on Biodiversity and Ecosystem Services. And he also serves as a review editor in Frontiers in Marine Science and a member of the editorial board of the Journal of Biodiversity and Biotechnology. Dr. Anticamara got his PhD from the University of British Columbia, Resource Management and Environmental Studies, Environmental Studies. Okay, his research focus is on biodiversity and conservation, coral reef biodiversity, ecological effects of disturbance and management, ecology of parks and reserves, ecological function and restoration, fishing trends, impacts and management, climate change impacts on coastal ecosystems, and future scenarios of coastal biodiversity. His recent publications are on the emerging response options and scenarios of slow onset events related to climate change in Southeast Asia and spatial distribution in benthic macroinvertebrate functional feeding groups in tropical rivers. So without any further ado, we have here Dr. Jonathan Anticamara.
Hello, good afternoon, everyone. So, thanks for inviting me. Can you hear me okay?
Yes, sir, naririnig po.
Okay. So, thanks a lot. Again, good afternoon to everyone, and thank you for having me. I'll just need to have the presentation in full screen. Okay. Okay. So, anyway, thanks for the very long introduction, and good afternoon to all, to the participants and to the previous speaker, Dr. Pavia. So I work on coral reefs since 1997, and I've been diving across the entire country, spent thousands and thousands of hours surveying coral reefs from northern Philippines all the way to Tawi-Tawi. And most of the time, I drive from Manila to sometimes Mindanao, taking with me an assistant, my dive gears, my compressor, scuba tanks, sometimes cats, and we frequently visit many of the coral reefs here in Luzon. So before the pandemic, I go to Batangas 4 times to 6 times a year, and Zambales, Hundred Islands, Bulinaw. So I spend a lot of time. So I teach half of the semester, and then the other half I usually spend going out there in the ocean, surveying coral reefs. For the last almost 3 decades, I've been studying. I spent my life studying diversity of coral reefs. So I documented species richness of fish and their status. I focus a lot of my research on that, and then I have the data for corals, but I'm trying to hold off because I feel like I need to do more in terms of the molecular aspect. So I established a molecular lab about 3 years ago to start and understand and verify some of the species that I find problematic. But anyway, so today my talk will be about some of the works that I did trying to assess the status of the corals. And um, the findings that I have is that many reefs around the Philippines really struggle to survive, like Dr. Pavia said. I can show you some data for that. And I also attempted to do some growth of fragments, so trying to assess their growth and survival in blocks, for example, and try to monitor them for almost 8 years now. And in some places, I did not really see a lot of recruitment or larval settlement. So part of the reason why I am interested in coral development now, in addition to the genetics, is to really understand why over vast places in the entire Philippines, there are not a lot of recruitment. And I think that this, I can explain later the trend. But the fact that I need to survey the literature to really understand developmental biology of corals, the challenges that they're facing with the recent disturbances in the ocean from sedimentation, dynamite fishing, ocean acidification, etc. Okay, so half of my talk therefore will be about research that I accomplished, and the other half is like basically notes for myself as I embark into the study of coral developmental biology in order to understand why corals are not recruiting in many of the reefs that I study, and a lot of corals are dying. Okay, so these images that you see are some of the pictures that I took. I took thousands and thousands of pictures to identify species. I identified many species in the Philippines. So right now I have a thesis student that's trying to update the checklist of Philippine reef species, Scleractanian, because if you look at The records, most of the records, the work on the Menzo. For example, I'm taking care of his collection of specimens and many holotypes, but some of these are no longer valid based on worms. And not only that, there's a need to really kind of update all our identification of corals in the Philippines. And also, if you look at worms, many of the corals that are put in there actually do not have maps of where it is found in the Philippines. So when you search for the Philippines, sometimes only 30 corals will appear, when in fact, we have an estimated of around 500 to around 700 coral species in the Philippines, still to be validated. Okay, so what are corals? Uh, when you go around the Philippines and you go in many fishing communities and you ask people what are corals, then most people will tell you that corals are rocks. So the people in fishing communities, they treat corals as rocks. And in fact, there are in YouTube, I watched some of conservation videos on coral reefs in the Philippines, and some prominent coral conservation organizations have even proposed ideas and talk about corals being a both an animal, a plant, and a rock. And I really think sometimes that can be misleading because really corals are animals. Once you treat them as a rock, then you'll find that most people will actually pick up boulders of corals like you see this ones here, and they use it for building bridges or their houses, and they would even dislocate corals because they just think they're just pieces of rock. But that's because during daytime, most corals are hidden, the polyp at least are hidden, they're retracted. So when you see them, if they're very small polyp, then you will only see this structure, which is their skeleton. But at night or even during daytime, some species actually protrude their tentacles. And like these octocorals, which is the more elaborate version of the polyps of the Sperctanian corals, um, they actually are alive at night, so they move. So corals are animals, but they do have symbionts. Some of them, some don't have symbionts, and but majority of them have symbionts. So that is expressed in terms of their colors. So the different symbionts will be expressed in terms of the different colors of corals. Okay, so I'd like to establish that I think it's good when we talk about corals, especially Scleractanian corals, a member of a very big group, the phylum Nidaria, which includes all the jellyfish, the octocorals, etc., and are the basal the group to the Triploblast bilaterian. Okay, so there's a lot of interest in coral development because, for example, they don't have really the third layer tissue, but they have the mesoglea. So that's the jelly-like structure that kind of look like the third layer, but it's not really the third layer. So they're the transition between the diploblast and the actual triploblast. So they are the sister group. So a lot of people studying developmental bio of corals because they want to know how this development are actually related to higher bilaterian. And why are corals very important? So in my work, most of the things I did, I tried to account for what are the values of coral reefs in the Philippines, and most of these are expressed in terms of fisheries. A healthy coral reefs, when protected, most of them are all protected areas that are sort of the few remaining healthy corals that we can find in the Philippines because most are dead or overfished. So this one, for example, is in Tucson, Camiguin. It's a very well-enforced protected area. When you swim in this coral reef, you'll see lots of big fish. And you look at the water column, you'll find jacks and schools of them. You find on the crevices and ridges of the reefs, and you'll find large snappers and sweet lips, etc. This is not a common picture when you swim around the Philippines. Most places in the Philippines are really overfished. But the value of coral is not only in terms of fisheries. Of course, fisheries, food is very important to Filipinos. So a lot of um, communities in the Philippines are found in the coastal areas. A majority of those coastal areas, they're highly dependent on fishing for livelihood and food. Okay, so maybe in Samar or in Zambales or in some islands in Bohol, about 80 to 90% of the people who live there actually are just fishing. They have no other means of livelihood. So the degradation of corals has strong impact onto their lives and their economy. But beyond fisheries and livelihood and food, corals host a lot of biodiversity. So probably the highest diversity in terms of per unit area found in the entire planet. And these are usually in terms of the various invertebrates. So on land, there's a high diversity of taxonomic diversity of invertebrates, but most of these are dominated by insects. In coral reefs, there's actually a wide array of taxa. So most of the phylum that we find for invertebrates can be found on coral reefs. And there are taxa that are not found on land. So that means that coral reefs win in terms of diversity of various taxa. And without corals, many of this taxonomic diversity generally also disappears. Okay. So again, what are the value of coral reefs? So I spent some time trying to figure out how can you value coral reefs, because when you swim around the entire Philippines and you see dead corals, you feel it's such a waste, and you feel that the people actually, the Filipinos, how do they understand the value of coral reefs? Even fishers, because many fishers they don't really swim, they don't know corals well. They cast their nets, they throw their hook and lines, and they hope for fish to bite their hooks or get into their nets. But they don't really see what's happening underwater. So except for those spear fishers. So majority of Filipinos do not really understand the interconnection between the value of the reefs, how reefs produce all these values, and the status of the reefs. But when we accounted for all the possible ways we can account for values of coral reefs, actually in the Philippines we derive around 40 billion US every year from coral reefs. And these are underestimates because, for example, the fisheries data that we use for this estimate, the prices of fish is really very low. So the government prices in their database would present something like a kilo of fish is around 80 pesos. But when you go to the market, the actual price for the kilo of fish is actually 3 times that. So I think that our estimate is really an underestimate, and there's really limited data. So we kind of extrapolate for some. But overall, we find that it's about 40 billion US, or sometimes a range, depending on what you're looking. If you look at the fisheries, you can get up to this much per hectare per year. And tourism really is also high. Most of the high value reefs are found in the central Philippines, because this is where a lot of tourism and a lot of fisheries are also being studied and documented. Okay. So what's the problem? The problem is that a lot of reefs in the Philippines have been declared degraded since around the 1980s. And if you know the late Dr. Gomez, he published paper stating that only 5% of corals in the Philippines, in terms of his sampling stations, are in healthy conditions. And although for me this is relatively difficult to interpret statistically, because I started asking question, does that mean that when you go around the Philippines, only 5% is left, or only 5% of the entire coral reef is healthy? But when you go in many of these places, it doesn't look like that. You'll find patches of reefs that are healthy, patches that are not. So I think sometimes it's there's a need to quantify how the reefs are degraded, and I think that even the most degraded, at least in Luzon, even the most degraded reefs sometimes have patches of healthy reefs. But in some places, like in the Visayas in summer, when you swim, you'll find something like this. You see vast expanse of reefs that are totally dead, because there's a lot of, even until now, there's a lot of dynamite fishes. So you see all these big boulders. These are big porrites that were toppled down by really large dynamites. So sometimes it's a gallon of dynamites being thrown in the ocean on a daily basis. So that means that this place not only silted, there's dynamite fishing, there's overfishing, and you can see there's almost no reef coming back, no corals, no larvae, no baby corals coming back. And so these are the status of some of the reefs, and I'll show you later the average status of all the reefs that I surveyed. But even the healthy reefs, like the one on the right, you can see that there are many of them that still have high coral cover, high diversity of corals, but almost no fish left. If there are fish left, they are the tiny ones. So the big fish are gone. Okay. So this is the data that we gathered from all the reports, and we those that have a coordinates, and we try to map them, and it shows that we use different colors to represent the percentage live coral cover. In the red one is less than 25, the yellow one is between 25 and 50, The blue one is between 50 and 75, and the green one should be 75 and 100. Even in there's a few data from 1970s and 1980s. In the 1990s to 2000, you'll see already that many reefs in the Philippines, when surveyed, are in red. So that means only about less than 25% live coral cover are seen. Okay, so we're seeing at that time. And this trend continues from 2000 to 2012 in terms of the data we look at. Okay, based on my entire survey, so I went through around the Philippines, as I said, and I surveyed many transects, hundreds of transects. So for every municipality, I surveyed at least 3 to 6, sometimes 8 islands, and within each island, I surveyed 3 to around 6 transects. Each transect is standardized around 20 meters. I took videos of all the species I found. I took quadrat videos of all the transect, 5 for each transect, and I analyzed this, and I plotted the frequency distribution in terms of percentage coral cover and also the species richness. I found that in all these places that I went and dove, you can find on average around 16 or 14 to around 16 species per transect. And there are places where I can find around 40 to 45 species per transect, but there are also species that are too degraded, that they have very few species left, and also very low coral cover. So I think that maybe what we need is really to try to understand the scale um, and the effect of scale in terms of how we quantify um, the status of coral reefs. And I think it's best to represent the data in terms of actual points that we survey and to standardize the survey so that we can track what will happen to these reefs over time. If we base it from previous studies, the methods vary. So sometimes the change might be difficult to detect because of the changes also in the methodology. Okay. But what's happening is that although some of these reefs are still relatively healthy, what I found is that many of these reefs no longer have fish left. So there's still high diversity of fish, but many of these species are tiny fish species. So they're in terms of their abundance, there are very few of them left, and their biomass is also very low because many of them are small fishes, except for the well-protected areas. So if you want to swim with me and go around the Philippines, what we'll find is that a lot of reefs are becoming either dead or empty. And the consequences of such um, degradation is that many of the fisheries have collapsed. So if you look at major fisheries in the Philippines, many of them have already declined for around 1980s and never came back up. And now we're pushing all the rest of the other relatively healthy, like Palawan, and now it's starting to decline. So most of the fisheries are mobile. They don't fish in one municipality or one barangay. They fish everywhere. And when that place is depleted, they move around. So now the fisheries that are fishing in Palawan are actually fisheries from Bohol because Bohol is too depleted. So this kind of serial depletion is occurring in the Philippines and with really limited regulation. So what happens is that fisheries just move around to deplete the next possible place to overfish. The real consequences of this is extreme poverty. If you try to go around the Philippines and you see these fishing communities, many of them are really in extreme poverty. With what they do is that they spend sometimes 4 liters on gasoline, and the gasoline is more expensive in islands. Sometimes it can go up to 60 to 80 per liter. They go out there and they spend that gasoline and they come back, they catch only 1 kilogram of fish. They sell that, they lost money, they are in debt. And it's to them, the fishing has become like a gamble. Sometimes you get a lot, but in most cases, you get nothing. So therefore, the poverty can be explained by the fact that the disappearance of fish and the degradation of coral reefs are causing the decline in their fish catch and therefore their income. And because most of these places have high population and a high number of children, then the poverty is very difficult to break. So what's the solution? I spent my entire PhD working in Bohol, living in islands, trying to help people recover some of the reefs. And at that time, the solution was really protected areas. So protect some areas. Do not let fishing happen in those areas so that fish can grow, they can reproduce, they can see the fishing grounds. And my research for my PhD showed that indeed this can recover, and sometimes it can be very fast. So this complicated graph is actually just to show that if you look at dominance of patterns of fish in terms of abundance of biomass, there will be a switch when you look at from a fish site and compare that to an old site. So there's a switch in terms of the blue and the red. And that means that the red one now is higher for the old protected areas, meaning that these places now are dominated by large-bodied fish. That's why the biomass curve is higher than the abundance curve, whereas the younger protected areas and fishing grounds actually are dominated by small fish, and therefore the abundance curve is higher. Okay, what's the other solution? So this year I published with one of my MS student that even inside the protected areas that we look at in the entire Philippines, the corals haven't recovered. And this is also something that I have been observing. So the fish have recovered in protected areas, but the corals are not. For some places, places that are highly sedimented, places that have a lot of river, or so that means that there's a lot of inflow of fresh water during typhoon or other seasons, rainy seasons. So what that means is that maybe protected area is good, and most protected areas in the Philippines are very small. So I started moving away my research into other methodologies that we can recover coral reefs, but can actually recover even the fishing grounds. So not just the protected areas. So I started experimenting on reattachment of coral fragments. So initially, I established blocks in Luzon, Batangas, and the islands, etc. These are standardized blocks of 50 by 60, and I put a lot of them. I attach the coral fragments and monitor their growth and survival over time. So now it's almost 8 years, running 9, that I go and visit these blocks, and I can show you that some species can grow so fast. So for example, this block, I reattached the fragments in 2014 April. By November 2014, this block was almost covered already by this species. But it really depends on the species. So so what I did is I tried to do this for many species. So I'm preparing another paper on the comparison of over a long term of growth rate and survival of coral fragments on these blocks. But what I also noticed is that there are few recruitments, but very few, and in some places there are none in terms of coral larvae on this block. So it begs the question, and that's kind of the question that leads me to explore more in terms of the coral development and development biology of strengthening corals. Okay. So anyway, so this is why I'm kind of shifting my research to both molecular and now developmental bio. So I foresee myself in the next 3 to 5 years spending maybe a lot of dive nights trying to observe how corals reproduce across comparing across many species. Okay, and so in preparation for this, I look at the literature, and in fact, there's very few publications on coral development. So when you look at Scopus, for example, I search in for November 2021, you can find around 400, and it's rising, it's increasing. But if you actually look at the details of each of these publications, many of them do not actually talk about gametogenesis or the development of planula. And corals are complicated organisms. I'll explain later in terms of their reproduction. And it's very difficult to observe for some, especially the ones that are brooding. Okay, so I think that there's a lot of challenges that are being recognized by developmental biologists, but it doesn't mean that we cannot increase the our understanding. So that means that we just need to spend more time studying. The other trend that I saw is that very few research are really done in the Philippines in terms of coral development biology. I saw a few, less than 10, that look at patterns of spawning and also gametogenesis, but haven't really looked at the development and only look at very few species, limited number of species, I think less than 10 species. So majority of research are actually done in Australia and the US. Okay, so I think that there's therefore a lot of opportunities for us to contribute in terms of developmental biology for corals, since we have so many species, probably more than any other countries in the world. A lot of this are done in James Cook. So these are the universities that produce a lot of paper on coral development. And again, in terms of country, you don't find Philippines because our publication is less than 10 on coral development. and a lot of research were funded by this group, but you don't find a lot of funding from the Philippines on coral development biology. So maybe this is one area that hopefully funding agency in the Philippines can also be, given the value of coral reefs like 40 billion US dollar a year, and letting that go, without spending money on it, and causing millions and millions of Filipinos suffering from lack of access to livelihood and food is not a good investment strategy. But I don't think at the moment the Philippines is investing a lot or enough for corals. Okay, so what are the trends? I have about 5 minutes to go through this. When I try to find papers that I can standardize. So basically, I can put into Excel and create some columns where I can compare across the literature in terms of species, how they've been studied in terms of gametogenesis or various developmental stages of the corals from the first division up to the planula stage to the settlement and to up to the metamorphosis into an attached coral. Okay, so there are a limited number of species have been studied based on the literature that I surveyed. It's still incomplete because I still have few papers to skim through. The problem with literature review, for example, if you rely on Scopus alone, you will not find all the papers that you need. So you in addition to that, you really need to search more one by one. Okay, but the best thing about